Since we're talking about human physiology in AP 120, it's important you know how these concepts of osmosis are super important to medicine. Every time a doctor or a nurse gives a patient IV fluids, those fluids need to be isotonic to the patient's cells. If a doctor or a nurse were to give a patient hypotonic or hypertonic fluid in that IV line, then it would damage all of the cells that line the blood vessel and also would damage the patient's red blood cells. Why is that? So remember that these words, iso, hypo, hypertonic, these are all comparison words. And now, if I tell you that the doctor has given the patient hypotonic fluids, you need to ask yourself, hypotonic compared to what? And the answer is hypotonic compared to what's inside of a patient's cells. That would mean that compared to the fluid, the patient's cells are hypertonic, right? Okay. So if the patient's inside the patient's cell and outside the patient's cell, if those two things are isotonic to each other, then the cell is going to be happy because there will not be any net movement of water by the process of osmosis. However, if you take a patient's red blood cells and put them into water, water is very hypotonic compared to the inside of a red blood cell. Water wants to move from hypo into hyper. And so water would go into the cell, into the cell, into the cell, till the cell gets so filled with water, it just pops. Here's how I remember that. I remember that a glass of water is hypotonic, and a raisin, since it has almost no water and it's almost completely solute sugar, if I drop the raisin into water, what happens? Well, the raisin fills up with water, right? The reason that happens is osmosis. Now you can bore people with the story about raisins and water, but it's an easy way to remember it. What would happen if I put a red blood cell into a hypotonic solution? Hypo is water. What happens if I drop a raisin in water? It fills up with water. You won't get that one wrong, right? Now, the opposite would happen if the doctor or nurse gave the patient a hypertonic solution into a blood vessel in the IV fluids. That would mean the water would want to leave the cell, and then the cell would shrivel up. And this is actually what it would look like. A happy red blood cell looks quite a bit like that. And as the water leaves and they get shriveled up, they start to look like that and that. And that has got a name. It is called crenation. When the cell gets so full it pops, it is called lysis or lysing. When it shrivels up, it is called crenation. Great. Now, there's more. We still are talking about passive transport when we talk about the movement of molecules like glucose into the cell. We've just finished talking about the endocrine system, probably in lecture, right? And in the endocrine system, you learned that when insulin is present, glucose is able to get into the cell. And how did that happen? You learned that when, this looks very chaotic. You learned that when, I do not know why. You learned that when insulin is present, that special transport proteins called glucose transporters get put into the cell membrane. And when those are in the cell membrane, then glucose is able to enter the cell through the process of facilitated diffusion, right? Facilitated diffusion. When glucose is concentrated out here, not very much inside, insulin comes along and says, hey, cell, please put glucose transport proteins into your membrane. Then the glucose will enter there and it will want to go into the cell because glucose still wants to be able to diffuse. Facilitated diffusion is still a type of diffusion, so it's still a type of passive transport and does not require cellular energy. But now that we're going to be talking about what nerve cells do, the concepts of facilitated diffusion and active transport become even more important. Let's go back here. As you're learning about what happens in the cell membrane of nerve cells, you will learn that there are two kinds of channels. One is called a potassium leakage channel, and the other is called a voltage-gated sodium channel. These are both facilitated diffusion channels. Facilitated diffusion channels generally are very specific. In other words, this channel that let glucose go through will not let sodium or potassium go through. The potassium leakage channel, you will learn, allows potassium to leak out of nerve cells down their concentration gradients. Remember, down the concentration gradient means from wherever the potassium is in higher concentration to wherever it's in lower concentration. That's the concentration gradient. The sodium, the voltage-gated sodium channel, will allow sodium to enter the cell at a critical moment in what a nerve cell does. But it still is facilitated diffusion because it's passive and it doesn't require any energy. But there's more, okay? We now need to talk about the active mechanisms. The active mechanisms are the things that the cell will do where it uses its own cellular energy to move molecules. Active transport requires the cell to use energy, ATP, and it is the only way to move a substance from an area of lower concentration to an area of higher concentration. For 3.5 videos, we've been talking about passive transport. We've been talking about how clever ways the cell can use the power of diffusion going from higher to lower concentration to move substances so that the cell doesn't have to pay. But particularly when we're talking about what nerve cells do, nerve cells will deliberately move sodium against its concentration gradient only to allow it to re-enter the cell by facilitated diffusion. So when we talk about the sodium-potassium pumps in lecture, when we're talking about what a nerve cell does, it is specifically using active transport. It is forcing sodium and potassium to go opposite their concentration gradients, forcing sodium to move from where there's not very much to where there's already a lot, only to open up voltage-gated sodium channels and allow it to come rushing back in. I'm going to be talking about that specifically when we're in the neurophysiology lectures, so I want you to connect it to what we're learning in lab this week. Okay. Now, this type of active transport that's usually just referred to as active transport moves molecules one or a few at a time. When we're talking about the sodium potassium pump, it moves 5 ions at a time. But, yeah, there you go. But there also is a way that a cell can move like a whole passel of things all at once, and that is by processes called endocytosis and exocytosis. First of all, in general, endocytosis is bringing things into the cell, and exocytosis is spitting things out of the cell. When we're talking about this moving a whole bunch at the same time, endocytosis and exocytosis require the cell membrane of the cell to move big packages of zillions of molecules all at the same time. Okay, I don't know what a zillion is, but lots, like hundreds of thousands, all at the same time. Exo refers to outside or to the outside in medical terminology, just like the exit to the room takes you out of the room. And endo is always the opposite. All righty? So let's talk about another way of bringing things into the cell. By endocytosis. With endocytosis, the cell wraps its membrane around hundreds of thousands of molecules and brings them inside. There are 2 types of endocytosis. When the cell wraps itself around a particle of something, like some food, that is called phagocytosis. It's kind of like when the cell eats. Human cells don't do that very much, but I will be showing you what happens when a white blood cell eats a bacteria. That would be a good example of phagocytosis, which is a type of endocytosis. Now, there are more cells in the human body that drink, and endocytosis, when the cell membrane is wrapped around a droplet of fluid, is called pinocytosis. Now, the opposite, when a cell spits out a whole bunch of molecules at once, that happens all the time in human cells. And it is known as exocytosis. And whenever I am saying that cells secrete something like beta islet cells of the pancreas secrete insulin, I am actually talking about the process of exocytosis of insulin by beta islet cells in the pancreas. All right, good, we got it all done. But particularly keep these concepts in mind as we're discussing what happens in nerve cell physiology.